Everyone. This week we're going to talk about microbial metabolism. In this video, we're going to discuss the differences between aerobic, anaerobic respiration, and also fermentation. We're going to go ahead and start talking about aerobic respiration. So some bacteria out there are capable of going, through aerobic respiration. If they go through aerobic respiration, they're going to go through the 3 cycles that we've already talked about in the previous video. They're going to go through glycolysis step, and then they're going to go through the Krebs cycle, and then they're going to go through the electron transport chain. The glycolysis step is going to yield about 2 ATP molecules. The Krebs cycle yields also about 2 ATP molecules, and then the electron transport chain is going to yield the most amount of the ATP. Now, aerobic respiration is going to yield a total of about 36 to 38 ATP molecules. Now, aerobic respiration, oxygen is definitely used during the electron transport chain as the final electron acceptor, because during the ETS, or the electron transport, electrons are going to be transported. And then finally, as those electrons are being transported within the ETC cycle, oxygen goes ahead and grabs all of these electrons, and oxygen acts as the final electron acceptor. Now we're going to go ahead and talk about glycolysis just in a bit more detail, because it turns out that there are alternative ways of glycolysis that bacteria can use. Okay, so not all bacteria can go through the exact same glycolysis pathway. So let's go ahead and take a look at glycolysis together. So glycolysis is the idea of breaking down sugars, okay, to make ATP, to also produce pyruvic acid, okay, and some other byproducts. But the thing is, it doesn't always have to be carbohydrate metabolism. So bacteria can also go through like the glycolysis pathway, but not through metabolism of glucose per se, but they can go ahead and metabolize things like proteins, things like lipids, okay? And the products would still be able to kind of like, go through, kind of enter into that glycolysis pathway, and glycolysis would still be able to take place. So again, this kind of image is showing you that it doesn't always have to be the metabolism of glucose. Bacteria can metabolize things like lipids and proteins and still kind of go through that glycolysis pathway, okay? Now, the glycolysis pathway in both actually animals and for most bacteria, that glycolysis pathway in bacteria, we refer to it as the EMP pathway, or the Embden-Meyerhof pathway. And that is the most common glycolytic pathway that bacteria would actually go through. A note is that glycolysis happening in bacteria, it happens in the cytoplasm of the bacteria. As I said, it yields a net amount of 2 ATP. It yields also some pyruvic acid, and then some other byproducts like NADH, etc. And NADH, by the way, is just a compound that's going to hold a bunch of electrons, okay, that are going to be used in the ETC cycle, okay. Now, so this is the most common glycolytic pathway, okay, called the EMP pathway. Now, glycolysis itself doesn't need any oxygen, okay? So, glycolysis, the pathway of glycolysis doesn't need any oxygen, doesn't use any oxygen, okay? But when we talk about the whole entire process of like aerobic respiration, yes, to go through aerobic respiration, oxygen is going to be needed to go through the Krebs cycle and the ETC cycle. But just the step of glycolysis itself doesn't need oxygen to go through. And as I said, that the common glycolytic pathway for most bacteria out there is the EMP pathway. Now, some bacteria actually out there use an alternative glycolytic pathway. So they go through other glycolytic pathways, such as the ED pathway and the triple P pathway. So the ED pathway, that's, as I said, it's kind of like an alternative pathway to glycolysis, and it's called the Ender-Dodrif pathway. Glucose is still kind of, again, the main kind of nutrient being used, and glucose is usually converted to ethanol, okay? And it usually yields 1 ATP instead of 2, okay, as the EMP pathway. So there are some differences, of course, as you can see in the products that are going to be released if the bacteria goes through this pathway. Bacteria such as, Pseudomonas bacteria, Rhizobium, and Agrobacterium, they are able to go through the ED pathway, okay? And some bacteria out there, such as these three groups right here, they are only capable of going through the ED pathway. They don't go through any other pathway, okay? But there are actually some bacteria out there, like E. coli. They can actually have the ability to go through the ED pathway or the EMP pathway. So they can, go through either if they want. Now, there is another pathway other than the ED, and it's called the triple P pathway. Again, that's just another alternative, okay, to glycolysis. And the triple P pathway, it's usually used for the biosynthesis of nucleotides and amino acids. And so this pathway, why would some bacteria choose to go through this pathway instead? Most likely, if there is a need, okay, for bacteria cells to make nucleic acids or to make proteins, then they're going to favor and actually go through this pathway, okay? Again, if the bacteria is in need of nucleic acids and protein synthesis, it will choose to go through this one right here, okay? So one more time, aerobic respiration, the bacteria is going to go through glycolysis, but as I said, there are alternative, when we usually talk about glycolysis in bacteria, the pathway is the EMP, right? Because this is what most bacteria go through. But as we looked at together and we kind of seen together that there are alternative pathways to glycolysis, bacteria could choose to go through the triple P pathway or they can go through the ED pathway, okay? But whichever pathway they go ahead and they go through, eventually after that, they're going to go through the Krebs cycle and then they're going to go through the electron transport chain where oxygen is going to be used as the final electron, acceptor, okay? Now, if we take a look at the electron transport chain, and again, we've talked about it in the last video, but this is just to kind of show you one more time, during the electron transport chain, which is going to take place in the plasma membrane of the bacteria, electrons, okay, that have been collected throughout the, glycolysis or the alternative pathways and throughout the Krebs cycle, all of these electrons that have been collected, those electrons are going to be transported, okay, through the ETC pathway. And as those electrons are going to be transported, a bunch of energy is going to be produced, okay? Now, in the ETC cycle, there are those kind of electron carriers that are basically going to help the electrons get transported, all the way down. Now, these electron carriers are called cytochromes. Now, they come in varieties, so there are cytochrome oxidases. And it turns out that there are some bacteria out there that can actually use different oxidases out there. And we use that to our advantage to actually differentiate between different bacteria. So there's a test, there's a biochemical test called oxidase test, and we can determine, whether it's an oxidase positive test or a negative test, meaning we can determine which bacteria out there have the capability of producing oxidase or not producing oxidase. So some bacteria out there, such as the gram-negative Pseudomonas aeruginosa bacteria, and a gram-negative bacteria called Vibrio cholera. So Vibrio cholera and Pseudomonas aeruginosa bacteria are actually capable of producing that oxidase, okay, and using this, cytochrome oxidase. And so when you do the oxidase test for those two bacteria, it will actually be positive. Bacteria such as E. coli, um, E. coli are going to test negative for the oxidase test because it turns out that E. coli, even though they can go through the ETC cycle and everything, E. coli do not really use oxidase, okay? And so when you do the oxidase biochemical test, it will turn out to be negative, okay? They produce different types of electron carriers, okay, that you're seeing right here, but not oxidase, okay? So we use that oxidase test to help us differentiate between bacteria that do use oxidase during their ETC cycle and bacteria that don't use oxidase during their ETC cycle. But bacteria that go through aerobic respiration and they go through the ETC, chain, at the end of the day, they're going to use oxygen as their final electron acceptor, okay? And that's going to be a major difference between bacteria that go through aerobic respiration and bacteria that go through anaerobic respiration. Bacteria that go through aerobic respiration, when they are going through their ETC, they use oxygen as their final electron acceptor. Those that are going to go through anaerobic respiration, they don't use oxygen as their final acceptor. They use things like nitrate and sulfate, okay, as their electron acceptors. So we're going to move on to the anaerobic respiration and talk about it in a bit more detail. So first of all, what are some reasons why bacteria would go through an anaerobic respiration versus aerobic? So some reasons why bacteria could choose to go through anaerobic respiration could be that, first of all, maybe the cell is lacking a sufficient amount of oxygen. So there isn't really enough oxygen for the bacteria to go through aerobic respiration. So if there isn't enough oxygen, the bacteria is going to go through anaerobic respiration. Okay, so maybe this is one, reason. Okay, there isn't enough oxygen. Or it could be now, when cells go through aerobic respiration, sometimes what happens is they produce some damaging oxygen like byproducts such as hydrogen peroxide. And hydrogen peroxide for bacteria is actually toxic. So those that go through aerobic respiration and these harmful byproducts such as hydrogen peroxide is released, they must have enzymes that will help break down these toxic byproducts. If the bacteria lack the production of such enzymes that will help them get rid of these toxic byproducts, then they won't be able to go through aerobic respiration and they will choose to go through anaerobic respiration. Okay. Another reason could be that, maybe they don't have the enzymes, okay? They are not capable of producing enzymes that will help them go through the ETC cycle, okay? And so one more time, they can't go through the aerobic respiration. Or maybe they are not, again, they're not making these electron carriers, okay, that will help transfer, the electrons down the ETC cycle, so again, therefore they can't go through aerobic respiration. So as you can see, there are multiple reasons, okay, why bacteria might, go through anaerobic respiration versus going through aerobic respiration. Now, if bacteria go through anaerobic respiration, okay, they will produce around, 32 to 36 ATP molecules total, okay? And those bacteria that will go through anaerobic respiration, they're still going to go through, the glycolytic pathway or, as I said, some alternative pathways like the triple P or the EZ pathway. They're still going to go through the Krebs cycle, the electron transport, but the thing is that when they go through the ETC, okay, they're not going to use oxygen. Okay, they're not going to use oxygen as their final electron acceptor. They are actually going to use things like nitrates, sulfate, okay, as their final electron acceptor, okay? Now, are there some bacteria out there that can actually go through both? Okay, yes, there are. So, bacteria such as E. coli can actually switch, okay, between aerobic respiration and anaerobic respiration. So, if E. coli is encountering a condition where there isn't enough oxygen, okay, in the environment, then E. coli is going to go through anaerobic respiration versus aerobic respiration. And when they go through anaerobic respiration, they can actually have the ability to use things like nitrate as their final electron acceptor, okay? And you can see, though, that when bacteria go through anaerobic respiration, the amount of ATP that they're going to produce is going to be less compared to if they go through aerobic respiration. So that's why, sometimes when we talk about bacteria and we say that those bacteria are facultative anaerobes, meaning that those bacteria have the capability of, growing in conditions that have high oxygen, and they can also grow in conditions that have low oxygen, then, this kind of tells us that these bacteria have the ability to switch between aerobic and anaerobic respiration. But they will always, if we talk about facultative anaerobes, they prefer aerobic respiration because, as you can see, more ATP will be produced via the aerobic respiration versus anaerobic respiration, okay? Now, some bacteria, though, are not able to go through aerobic respiration or anaerobic, okay? So they go through something called fermentation, okay? And with fermentation, those bacteria are only going to go through, the glycolysis pathway or the alternative, glycolytic pathways, but they're not going to go through the Krebs or the ETC, okay? And when bacteria go through fermentation, the net product of ATP that they are going to produce is around 2 ATP. So as you can see, huge difference, okay, if bacteria go through fermentation in terms of the ATP production. The ATP production is going to be way low, and it's going to be around 2. And actually, those 2 ATPs are due to going, through the glycolytic pathway, okay, because they're not going through the Krebs or the ETC, okay. Now, some bacteria, for example, such as Streptococcus bacteria, they are going to go through the fermentation process. So bacteria such as Streptococcus, they can't go through aerobic or anaerobic respiration. They cannot, they can't go through these type of respirations, even in the presence of oxygen. So even if they were given oxygen, they're still not going to be able to go through the respiration processes. They're going to go through the fermentation process. Okay. Now, why are those bacteria that go through fermentation, why aren't they capable of going through these 2 types of respiration processes? Well, maybe one reason is they don't have the enzymes that would help them go through the Krebs cycle or the ETC cycle. Okay, so they don't have the enzymes that will allow them to go through these two cycles right here. So again, they can't go through the Krebs, they can't go through the ETC. So they're only then capable of going through fermentation, since fermentation, doesn't have the Krebs or the ETC. Maybe another reason is they don't have um, the enzymes, okay, to help them make some of these electron carriers, or, to have the final electron acceptors. So therefore, again, they can't go through the ETC. So those bacteria would go only through the fermentation process. Now, with the fermentation process, of course, those bacteria will not use oxygen, okay? So no oxygen at all would be produced. They don't go through the Krebs or the ETC because, as I said, they lack the enzymes that will help them go through those, processes. And the amount of ATP that will be produced if they go through fermentation is really low compared to the other two processes. So only 2 ATPs will be will be produced. Now, so after they go through, let's say, the glycolysis step, right, and they produce pyruvic acid, now the products that will come out through fermentation could be different between different bacteria strains. So this kind of image is showing you that some bacteria, when they go through fermentation, the main product that they produce is lactic acid. Now, some bacteria out there, when they go through the fermentation process, they actually take the pyruvic acid and they turn it into alcohol. Okay, and they can also produce some carbon dioxide as well. And there are other pathways too. I mean, this is just kind of showing you how some bacteria can produce lactic acid and how some bacteria produce alcohol. Are there any other pathways that could take place and therefore the products might be different than lactic acid and alcohol? Yes. Okay. So let me kind of show you a table and show you like other products if bacteria go through the fermentation process. So right here, this is kind of showing you that, okay, the bacteria would go through the glycolysis pathway and they would produce the pyruvic acid. Now, once the pyruvic acid is produced, though, depending on the strain of the bacteria that we are talking about, different products can be produced, okay? And this table also shows you yeast. So yeast is a fungus. So when yeast also go through fermentation, it's going to shows you the product for yeast is going to be ethanol and CO2. And that's why yeast is used in the production of beers, right? Because they use the yeast fermentation product because it's going to produce alcohol, as you guys can see, to help them make beer. Yeast is also used to make bread, okay? Because when you put yeast in your dough, uh, CO2 is going to be produced, carbon dioxide, which is a gas. And so when you add yeast to your dough is going to puff because of that carbon dioxide that's being produced, okay? Versus if you don't put yeast in your dough, the bread comes out super flat, okay? So we humans are kind of using, these different fermentation products produced by bacteria or by yeast, to our advantage, to help us make, as I said, beer, to help us make bread, and then to also kind of help us make other fermented products like pickles and sauerkraut, etc., cheese even, okay? So if you guys take a look right here, bacteria such as Streptococcus and Lactobacillus and Bacillus, when they go through the fermentation process, they only produce lactic acid. Now, we actually use these bacteria, Streptococcus and Lactobacillus, when we are, when we want to make yogurt. Okay, so if you actually read the ingredients, okay, on like yogurt container, you're gonna see that it says it contains live bacteria such as Streptococcus, Lactobacillus, etc. And because the acid that they produce, right, when these bacteria are going through fermentation, the lactic acid, this acid actually denatures the proteins found in milk, and that makes, which causes the milk to solidify, right? So that's why we use it in yogurt production, and not just yogurt production. We actually use it also when we are trying to make cheese. Now, when lactic acid is the only fermentation product produced, we refer to this type of fermentation as homolactic fermentation. So let me kind of write it for you right here. So we refer to it as homolactic fermentation. And that's again when lactic acid is the only product being produced. So we refer to it as homolactic fermentation. Okay. Now, some bacteria out there, though, when they go through the fermentation process, they produce a mixture. Okay, as you guys can see, like they produce a mixture of products. They could produce like ethanol, lactic acid, acetic acid, some carbon dioxide. So as you can see, like it's a bunch of different products. And so we refer to this type of fermentation as heterolactic. Okay, so heterolactic, that would be the type of fermentation that would result in the production of various products, okay? Ethanol, acetic acid, and not all the time, like all of these products would have to be, produced. So sometimes bacteria would just produce like some alcohol and CO2 and, another type of acid. But if there are multiple products, as you can see, ethanol, lactic acid, CO2, etc., then that would be called a heterolactic fermentation. That's a T, by the way. Okay, so here. And again, we use these products, right, to help us like make things, as I said, like pickles, sauerkraut, etc. So we definitely, use bacteria, as I said, to our advantage to help us make, various food. Okay. And so this is the, the products of the, fermentation. As you can see, it's immense, and it also helps us when we are trying to differentiate between the types of bacteria that we have. So as you guys can see, like a Streptococcus bacteria right here would only make lactic acid versus, Escherichia, okay, and Salmonella. They can produce ethanol, lactic acid. They can also produce, as you guys can see, some gases. So we definitely do biochemical tests that would help us figure out, and differentiate between these different types of bacteria. We can do a biochemical test and figure out, whether the bacteria, when it goes through fermentation, whether it produces acid or not, whether it produces certain types of acids or not. Okay. So that was it, you guys, for this